All right, everyone. Thanks for coming out today. Super excited to start the AI Protein Seminar Series with Neil King. So Neil needs no introduction, but I'm going to talk anyways. Neil has been really a pioneer in protein design, and especially in the design of vaccines. So Neil began his scientific training as an undergraduate at Northwestern after growing up in the Midwest, and longtime Chiefs fan before it was cool. And then Neil went to graduate school at UCLA, joined Todd Yates's lab, where really started pioneering the design of self-assembling protein nanoparticles there. Afterwards, he joined David Baker's lab at University of Washington in Seattle, where he truly took it to another level, designing multi-component and single-component self-assembling nanomaterials during his postdoc before becoming a professor in 2017, translational investigator who started a lab a couple years earlier than that. Neil's also entrepreneurial, so he launched Icosavax, which is a company that he co-founded to develop protein-based nanoparticle vaccines. And Icosavax has actually quite a few programs that were acquired by AstraZeneca that are now in phase 3 and some accelerated approval status as well. And I believe these are the first ever medicines or vaccines, therapeutics, from the field of computational protein design that will be fully approved. So I would say a truly pioneering accomplishment there. And on top of that, Neil's just an awesome person. So with that, Neil, please take it away.
Awesome. Thanks. Super happy to be here. And what Chris didn't tell you is that he and I overlapped as postdocs in the Baker Lab, and we have a long string of failed summit attempts in the Cascade Mountains where we would get like so close to the top of the mountain, but then there'd be this like class 4 scramble, and we'd be like, maybe not. And then the other thing is, Chris has now developed a habit of going back and finishing those summits. So kudos. I still want to get Summit Chief. That one is like burning in my mind.
I'm game.
Okay, so want to talk to you today about learning-based vaccine design. And I probably don't need to tell you guys this, but we're in the middle of a technology-driven revolution in vaccine design. We've all been living through this together over the last few years. On the left side of the slide, I'm showing designed antigens. This is prefusion RSV-F from the VRC in 2013, where they stabilized the antigen in the prefusion conformation, which is the one targeted by the most potently neutralizing antibodies. Fast forward 10 years, that led to 3 licensed vaccines for RSV, the first ever vaccines for RSV. So proven model based on protein engineering. On the right side of the slide, right, we're in the hometown of Moderna here, so this is mRNA vaccines. We all saw what mRNA can do during the pandemic. So these are 2 examples of new technologies that have pushed forward vaccines. There are more than this. We'll be talking about nanoparticles and then glycans and glycobiology today. So my lab at the IPD is primarily a tech dev lab. We build new technologies. When we think about vaccines, we think of them as comprising 4 different technologies. You've got to have an antigen. You've got to have a piece of the bug that you're trying to protect against, right? I think we and others have shown that this works better if you put it on a nanoparticle, and we'll talk about why. You've got to pick up front your delivery modality. Are you going to make this thing as a recombinant protein, as an mRNA, whatever? And then you should be designing your molecule from day one for that delivery modality. And we'll talk about that. And then finally, and I think this is a huge area for growth, vaccines need to stimulate the immune system, right? That's what you're trying to do. And a lot of that is innate immune stimulation. And so we're developing protein design technologies for innate immune stimulation to make better vaccines. So the 2 major stories with sub-stories that we'll talk about today cover these 3 parts. We're not going to talk about antigen design today. Okay, so these are the stories. The first 3 are all about nanoparticle vaccines, so it's kind of like 3 chapters in a single story. And then the last one is about programming N-linked glycosylation. All right, so the reason we're going to talk about nanoparticle vaccines is because it's been known for decades that particulate immunogens elicit superior immune responses. This is because your immune system has evolved to detect and respond strongly to particulate immunogens. It does this in several ways. One way is antigen receptor clustering. So this is the B cell receptor on the surface of the B cell. And this receptor is activated by clustering, by multiple copies of the receptor being brought together physically. And that is directly sensing repetition in three-dimensional space, right? Viruses are symmetric, repetitive arrangements of proteins, and this is how your immune system senses and responds to them. Problem is, if you're developing a subunit vaccine where you take the antigen out of its native context, you often lose that repetition in three-dimensional space, you lose that strong activation, you lose a potent antibody response, right? So we wanted to come up with a technological solution to that problem. Another way that your immune system has evolved to detect and respond to particular immunogens is by trafficking them to the lymphatic system. So small proteins, like mini proteins, are rapidly distributed into the blood, whereas large particulate things that look like a virus, smell like a virus, drain very efficiently into the lymphatics, where they go to the draining lymph node, into B cell follicles, where the immune system can take a good look at them. So these are two major reasons. There's probably other stuff going on, but these are two major reasons that you might want to design particulate immunogens. So in ancient history, back in 2010, when I showed up in the Baker lab, I worked really closely with Will Scheffler to come up with this design process for generating new self-assembling proteins. And it's two fundamental steps. There's a docking step and there's a design step. In the docking step, back then, what we were doing was going and finding naturally occurring oligomeric protein building blocks, like this gray pentamer, this blue trimer, and then we would dock those pre-existing building blocks in some symmetric architecture. This happens to be icosahedral point group symmetry, but this could be any point group symmetry. It could be fibers, layers, crystals, whatever. And you see how those building blocks fit together in three-dimensional space. And then in the second step, you design an amino acid sequence that should encode that target structure. Here, because we were using naturally occurring protein building blocks, the only thing we had to design was this de novo protein-protein interface between them, right? So we'd come up with a new sequence for that interface, and if we get this right, that sequence should orient the two building blocks in just the right orientation so that it goes all the way around and closes and forms this nice protein soccer ball. So at the end of the day, what you get out of the computer is a hypothesis in two parts, a pair of amino acid sequences, and a three-dimensional assembly that those sequences are predicted to form. So we take those hypotheses into the lab and test them. These are 8 different icosahedral nanoparticles that Jacob Bale, a very talented graduate student that I worked with, designed. These are 120 subunit complexes. They're about the size of the smallest viruses, so like an AAV. And if you look at these 8, you can see that each particle has a different structure than its cousins, but within a single electron micrograph, every particle is an identical twin. So they're very monodisperse and homogeneous, which from a manufacturing perspective is really important. And then when we solve crystal structures of these so that we can evaluate at the level of individual atoms how accurate our design method was, we found in these two cases, and this is pretty typical for this approach, the RMSD over all 120 subunits was 26 angstroms. So Jacob put every atom where he said he would. Accuracy of about half the width of an oxygen atom. And that allows you to design these materials prospectively, right? The way that we turn these things into vaccines is we genetically fuse an antigen of interest to one or both of the nanoparticle subunits. So here I'm showing, this is our COVID vaccine. When SARS-CoV-2 broke out, we wanted to see if this tech could help fight the bug. So we teamed up with David Wiesler and Lexi Walls in the biochemistry department. Lexi solved the first structure of a coronavirus spike protein ever. So they're experts in coronavirus glycoprotein structure and function. And we put the RBD on the nanoparticle, so just the receptor binding domain that interacts with the host ACE2 receptor. We made spike particles as well, but the RBD particles actually worked better, so that's what we moved forward with. So here what you do is you have this genetic fusion of the RBD to one component of the nanoparticle. This happens to be the trimeric component. You produce that and the pentamer as two standard recombinant biologics. There's nothing special about them. And then once you have those, you mix them in a reaction vessel and they spontaneously assemble to form the icosahedral assembly. And now you've restored that repetition in three-dimensional space that is such good catnip for the immune system, right? And when we immunize mice with these particles, we see that we get very high levels of neutralizing antibodies, much higher than the prefusion stabilized spike trimer, and infinitely higher than just the monomeric RBD, which elicited no detectable neutralization. So this is the exact same antigen that we put on the particle, and so this increase in neutralizing antibody titer is the sort of performance enhancement that we've come to expect from the particles. When we shipped our immunized mice to Ralph Barrick's lab at the University of North Carolina so they could challenge them with their mouse-adapted SARS-CoV-2, we found that the particles completely prevented viral replication in vivo, whereas we saw a breakthrough at low dose of the spike trimer and no protection at all from the soluble monomeric RBD. And so skipping a couple years and a huge amount of work and going straight to the phase 3 data, the RBD nanoparticle was manufactured by our partners at SK Bioscience and compared against AstraZeneca's Vaxzevria. And the RBD nanoparticle induced 3-fold higher levels of neutralizing antibodies. And so this was licensed in South Korea, in the UK, and was given an emergency use listing from the WHO. And so this became the first licensed medicine from the field of computational protein design and clinically de-risked this nanoparticle vaccine platform. So that was an exciting moment for the field, but it was kind of disappointing for us because this vaccine did not make a tremendous impact because we were slow. It took 900 days from publication of the viral genome to licensure of this vaccine, which by historical standards is blazingly fast, but compared to mRNA, we were turtles, right? So mRNA was available in less than a year, and what really killed this vaccine is 2 weeks after this one came out, the bivalent mRNA boosters were licensed, and nobody wanted a monovalent vaccine anymore. So this vaccine didn't really save lives. So we wanted to see, can we make these things faster, right? We were killed by how long it takes to manufacture proteins. So this is where genetic delivery comes in. So again, you guys all know that mRNA vaccines are super fast to manufacture, and a big reason for this is it is a sequence invariant manufacturing process. No matter what the sequence is, it's the same manufacturing process every time. Whereas every protein, in contrast, is a unique and beautiful snowflake and requires a custom manufacturing process. So we wanted to know, can we marry the potency of the nanoparticle immunogen with the manufacturing speed of mRNA vaccines? And so what we're doing here is we call these things mRNA-launched nanoparticle vaccines. And I want to be really clear because sometimes I mess this up and people get confused. So here, this is a totally vanilla, totally standard mRNA. You know, N1 pseudomethyl uridine, modified nucleoside mRNA, completely standard. It's a totally vanilla LNP, Moderna BioNTech style LNP. Nothing special about those. The difference here is that the mRNA is encoding a different amino acid sequence. Instead of encoding membrane-anchored spike, we are encoding a secreted nanoparticle immunogen. So I've omitted the secretory pathway for clarity, but this it's a single polypeptide, so this is a homomeric assembly that is translocated into the ER, it assembles, and it is secreted out of the cell using the standard secretory pathway. So the cell spits out these nanoparticles, which drain to the draining lymph node and do their nanoparticle thing. So Grace Hendricks is the grad student that took this on, and so we used the SARS-CoV-2 as a model system here, and we're like fastidious about protein quality at the IPD, and so she purified the particles that the cells were spitting out. She characterized the heck out of them, made sure that they are the exact same particles. They bind all the right monoclonal antibodies, they bind the receptor, et cetera, et cetera. So the particles are beautiful. And then when we go do an mRNA study, we compared against membrane-anchored spike in green, and we used the exact sequence of Pfizer BioNTech's vaccine, but we did the same mRNA and LNP formulations as ours, so you could do a head-to-head comparison. And what we found is that after a single immunization in mice, we got 28-fold higher levels of neutralizing antibodies from the mRNA-launched nanoparticle than we did from membrane-anchored spike. And we included in this study a secreted trimer that we call a non-assembling control. So this is almost the exact same protein as the particle. It's like 95% identical at the amino acid sequence level. It just doesn't have the interface that drives nanoparticle assembly. So this thing secretes at a trimer and gives you 11-fold lower levels of neutralizing antibodies than the mRNA-launched nanoparticle. So these two comparisons tell me that being a particle matters even if you're an mRNA vaccine. So that was, and then if you challenge these mice, all of the vaccines protect the mice against weight loss, but the mRNA-launched nanoparticle best prevents viral replication in vivo. So that was one example, but we wanted to see if we could do this again, because it's not truly a platform until it works every time. And so this is a second example using Rift Valley fever virus as a target. And here, Zach is a graduate student in the group that generated a stabilized version of the GN head domain, which is the receptor binding domain for Rift. So you can think of this as another RBD nanoparticle just for a different virus. And he made the 2-component protein nanoparticle version of this, and then the 1-component mRNA launched nanoparticle version of this. And here I'm showing data. This is just post-prime. So this is after a single shot in the mice. And what we saw is that after a single shot, the protein nanoparticles actually don't elicit much neutralization. If you give them a second shot, if you boost them, it goes up like crazy and you get super potent neutralization. But after a single shot, not much. Whereas the mRNA launched nanoparticles gave us really robust neutralization even after a single shot. Whereas again, membrane anchored in this case didn't really do anything at all. And then this is the full neutralization series over the course of the study. Sorry, the axes are different here. And so if you compare the actual titers, a second shot gives you a pretty significant boost. And now we're at extremely high neutralization titers, whereas again, membrane anchored isn't really doing much at all. And here we went down to 0.1 micrograms, which is like a relatively low dose for mRNA in mice. We need to go lower, obviously, and so that's what we're doing now. But in this study, even at 0.1 micrograms, we saw complete protection against lethal challenge from Rift from all of the mRNA-launched nanoparticle vaccines, whereas membrane-anchored, again, provided partial protection. So this is a second case where being a particle is better than membrane-anchored. Okay, so we've got protein nanoparticles, we've got mRNA-launched nanoparticles, but to be self-critical, everything that I've shown you so far is using this old approach that's really retrofitting. So what I've shown you to date is we would design particles, we would take whatever particle we could get, because it was so hard to design a new particle that we would just take anything, and then we'd come back later and ask if a particular antigen might fit onto a particular particle, right? So it really is a retrofitting approach. But that works, but it's not ideally what you would want to do. Ideally, you would say, okay, I think this constellation of orange antigen in panel D is going to give me the best vaccine response. So then what I'm going to do is go design a tailored nanoparticle to present exactly that constellation of antigen. And using the old approach that I showed you, this just wasn't possible. You just couldn't do it 3 years ago using Rosetta. But now we've got all these machine learning based methods from the Baker Lab and others, right? And now you can actually do this. So you guys all know this, the de novo design paradigm is you come up with a target backbone structure, and that backbone structure should encode whatever function you want. You then design an amino acid sequence that strongly encodes that target backbone structure, and then you use structure prediction to filter your designs and identify a subset that you're highly confident in that you want to go test in the lab. And for this, we're using tools developed at the IPD by my colleagues David Baker and Frank DeMeo. And so for backbone generation, we're using RF diffusion and its progeny. For sequence design, we're using protein and MPNN. And then for structure prediction, we use Rosetta Fold and AlphaFold. I mean, I'm sure you guys have all seen this, but I never tire of watching these diffusion movies. When this first came out, I called it Ouija board protein design, because it's like a protein just appears from the great beyond, right? And you're like, where'd it come from? And this is using these diffusion denoising probabilistic models that are similar to image generation algorithms online, right? So these things work by taking every picture of a cat on the internet, shoving it into a neural network, and then gradually adding noise until you get to pure Gaussian noise. Once the model has seen that, at inference time, it can do the opposite, right? You can give it pure Gaussian noise and say, make me a picture of a cat sitting on a table, and voila, you get that cat. So it works for cats, it works for protein. And so Cyrus is a graduate student in the lab who's been using these approaches to design customized antigen-tailored nanoparticles. And so first, we just wanted to know, can we design de novo nanoparticles at all? And so these are not antigen-tailored yet. It's very similar to the overall strategy that I showed you very early on in the talk. But the difference here is instead of using naturally occurring building blocks, now we're just diffusing these things de novo. And so you can make a de novo dimer, trimer, tetramer, whatever you want for the target structure that you're trying to build. Then you dock those de novo building blocks just like before. At the sequence design level, the one difference is we don't have a pre-existing sequence for these building blocks like we did before, so you have to design the sequence for the whole particle in a single shot using protein MPNN. And then again, we use structure prediction to filter these things down. And so this works. These are 6 particles that Cyrus generated using this approach. I think it was 39 de novo particles that he built for this paper. 2 tetrahedral, 2 octahedral, 2 icosahedral. And a couple things to point out here. So, you can get that same type of monodispersity that we had come to expect from our previous methods, beautiful particles by EM. But if you look at the backbone structures of each of these things, they're very, very different. And this reflects the free-form nature of these diffusion methods. Every time you push the diffusion button, you get a totally different answer. And this is actually really important because it means that you can sample a wide swath of backbone structure space. So that's really cool, and again, something we couldn't do before. We, of course, wanted to know what the accuracy of this method is at high resolution. And so our EM core at the IPD solved 3 cryo-EM structures. We have crystal structures as well, not shown here, of 3 of these assemblies, and we see that same accuracy that we've come to expect. So 1 to 2 angstrom RMSD over the entire complex, right? So Cyrus is now generating de novo every single atom and putting them right where he said he would. So yes, we can make de novo nanoparticles. Next question is, can we actually tailor them to a specific application? So nobody had yet scaffolded a motif, a functional motif, in a self-assembling de novo structure yet, and so that's what Cyrus did next. And so here, we took a flu antigen that we had designed a few years ago in the lab that we call trihead, which is sitting on top of this 3-helix bundle. And Cyrus took just the orange motif at the bottom of the 3-helix bundle, fed it into diffusion and said, build me a building block out of this, right? So what he's doing is he's burying the antigen in concrete or building the particle out of the antigen, right? From there, it's the same process. You dock the building blocks, you design a sequence, and if this works, because we have an antigen-derived motif sitting right there at the 3-fold, we should be able to just snap our antigen back on in the lab and it should just work. So first, Cyrus made the particles, screened them in E. coli, and this worked as well. So these are 5 different motif scaffolded particles that Cyrus generated. You can see the particle morphologies are quite distinct. The backbone structures, again, are quite distinct, but they all contain that same 3-helix bundle at the icosahedral 3-fold. These particles were slightly more heterogeneous than what we're used to, so we're working on refining that. But once you purify them, they're beautiful. Every particle looks just like every other. And then Cyrus added his flu antigen back on, and it turns out that it did just work. He was able to create this and secrete it from mammalian cells. The particle became more monodispersed when he put the antigen back on, which we found interesting. But beautiful particles, they bind the on-target antibody, they don't bind the off-target antibody. That one shows that the trihead is actually closed. It forms a native-like closed trimer. And a cryo-EM structure showed that the underlying nanoparticle is intact and the antigen is actually fairly rigidly displayed on this particle. It's not perfectly rigid, but it's not bad. And then we did just a shoot from the hip immunogenicity study with this first antigen-tailored de novo nanoparticle, and we compared it against one of our clinical candidates that's in phase 1, and that's the one in green. And we saw that we got the exact same immune response from this shoot from the hip de novo particle compared to the state-of-the-art that's in the clinic. So we feel good about that and think that we can only go up from there. So the trihead antigen we had already put on nanoparticles before, so that was a nice test case because we knew it was possible. But the real strength of this method is that it generalizes to other types of particles for which no current solutions exist. So the other antigen that you'd want to put in a flu vaccine is neuraminidase, and neuraminidase has C4 tetrameric symmetry. And there are no existing particles that are capable of displaying tetrameric antigens by genetic fusion, and so we wanted to build that tech. And so Annie is a second-year graduate student in the lab that took Cyrus's approach and built a series of octahedral nanoparticles built out of tetrameric building blocks that match the symmetry of the antigen. and those display NA, and then we've since shown that those can display other tetrameric antigens like henipavirus receptor binding proteins and things like that. So the approach generalizes and can generate tailored nanoparticle scaffolds even for antigens for which no solutions exist. So that's fun. So there are additional nanoparticle stories more recent that I haven't put in here, but that's kind of where we are at the moment. Okay, so switching gears, let's talk about innate immune stimulation, and we're going to do it through glycans. And there's going to be a bit of an introduction here just to lay out the rationale, because it's a little bit unusual. In-linked glycans are a post-translational modification. They're ubiquitous in eukaryotic cells, and they're attached during secretion in the secretory pathway. So it's a or sometimes a post-translational modification. We're going to be talking about in-linked glycans today. So those are attached to the side chain of asparagine residues. That's why they're called in-linked. And every glycan is born as an oligomannose glycan and then is changed into a complex glycan. We'll talk about that. And most glycoproteins, and certainly most human glycoproteins, display complex glycans. So this is what that looks like in a little more detail. So you get your translocating polypeptide that goes into the ER lumen, and then OST, oligosaccharyl transferase, attaches this high mannose glycan, and every glycan is born as this exact same high mannose glycan. That's actually conserved throughout eukaryotes. That's not just mammalian. What happens after that is this long series of enzymatic transformations as the protein goes through the secretory pathway from the ER to the Golgi and outside of the cell, where a very complicated series of enzyme acts on this thing and ends up giving you a complex set of different types of glycans constructed from different types of sugars. So if you look at it, you can see that the mannose is trimmed down, actually, to get down to man 5, and then other types of sugars are added back on to get you to these complex glycans. So what we wanted to do was throw in a monkey wrench right here. Can we halt this process at ER alpha minosidase 1 so that we never trim down that oligomannose glycan? The reason we wanted to do this is because we were inspired by viral glycoproteins. So viruses, enveloped viruses, use glycans for immune evasion. And the reason they do this is because if your protein doesn't have glycans, the whole peptidic surface can be bound by antibodies. But if you sugarcoat your protein antigen, the sugar is hard for antibodies to bind to, not impossible, but harder, and you mask the underlying peptidic surface, and that makes it harder for the host to generate neutralizing antibodies, right? So that's cool, but sometimes the virus goes too far. And it puts on so many glycans that some of them come out as these underprocessed oligomannose glycans. And the reason that happens is because these processing enzymes, and the cyan enzyme shown here is ER alpha minosidase 1. Those enzymes are sterically prohibited from processing the glycan in question because the neighboring glycans would clash, right? So the virus has gone too far. It's trying to mask its surface. It adds in all these glycans, but now you get these underprocessed high mannose glycans that come out. And basically what has happened is you've got a monkey wrench thrown in at this exact step, and the immune system pounces. So beautiful work from Daryl Irvin when he was still here at MIT showed that these high mannose glycans on these viral glycoprotein antigens are detected by the immune system, by mannose binding lectin and other types of lectins that sense these underprocessed glycans and say, that ain't right, I need to go after that, right? And what happens is MBL and other lectins will activate complement, opsonize the nanoparticle immunogen, and facilitate its trafficking into the draining lymph node where the immune system can take a good look at it. So this results in a better antibody response against the vaccine. And that's work shown here. If you knock out mannose binding lectin, if you immunize a mouse that doesn't have MBL or complement, you get a lower immune response against these monosylated nanoparticle immunogens. So kind of the heretical way that we're thinking about this is that high mannose glycans can be considered an adjuvant. So in the vaccine space, adjuvants are molecules that you add to your vaccine to potentiate the immune response. They tend to be crazy molecules like saponins derived from the bark of some tree in Chile, or some other thing derived from shark fins. They're like horrible to work with, horrible to formulate. There's a better way, right? And so we're thinking of high mannose glycans as adjuvants because they potentiate the immune response. Problem is, what if your antigen doesn't have glycans, right? The work that Daryl did with Bill and others on these, HIV nanoparticle immunogens, the HIV part had these high mannose glycans that I showed you a few slides ago. But what if you're making a vaccine against bacteria, against P. falciparum malaria? Those antigens don't have glycans, so you can't do this, right? So Jake was a postdoc in the lab a few years ago that collaborated with Daryl's group to investigate how this works in a little bit more detail. And what we did was we made a systematic series of glycosylated nanoparticles where we varied the valency, so the number or density of glycans per particle, and we exploited the two-component nature of our particles to do this. It's very easy to make a series of particles with 240, 120, 60, 12, 0 glycans. And we also modulated the glycan type. So the way that you do this, generally speaking, is you add this small molecule inhibitor of ER monosidase into your tissue culture medium. And then monosidase, again, you've thrown that monkey wrench in, it can't trim down those oligomannose glycans. So when you do this, you get particles out that have 100% oligomannose glycans on them. And again, we can control the number of those high mannose glycans. And if we don't add chitinizine to our tissue culture medium, we get mostly pink complex glycans with a small minority of oligomannose that comes from that steric crowding from having so many glycans right next to each other. So Daryl's group put these into mice and looked at localization of the vaccine in red and was looking at co-localization with CD35, which is a complement receptor that's a marker for B cell follicles. And what you can see is this density-dependent effect. It's particularly apparent at day 7 post-immunization, where you see good localization of the vaccine with B cell follicles with high glycan density, and it just goes down as you go to lower glycan density. So that's what you see in this bar plot. As you go down in glycan density, you lose that trafficking benefit. And over here, these are the complex glycan nanoparticles where you see no trafficking benefit at all, no matter what the valency is. So it's got to be high mannose glycans, and the more the better. This, again, results in a better immune response. So here we're measuring the antibody response against the computationally designed nanoparticle, and having the high mannose glycans improves the response compared to the native glycans. Okay, so high mannose is great. You can make it with chitinizine in the lab, right? So what's the problem? Problem is, once you get up to commercial scale vaccine manufacturing, chitinizine is just economically infeasible. You cannot do this at commercial scale. In mRNA vaccines, it's impossible. You can't encode chitinizine in an mRNA vaccine, right? So really what you would want to do to have a general purpose technology is to genetically encode these high mannose glycans. Now, one way to do this is you could just shove a bunch of glycans onto your protein and hope that you get this clashing from neighboring glycans. But, as I always tell people in my lab, and I'm sure Chris says something similar, protein design is all about one word: control, right? And I would say this is imprecise and messy. We would like to have more control than this. So as we were looking around trying to figure out how to do this, we came across this protein called uromodulin. It turns out there are very rare cases where naturally occurring proteins actually genetically encode high mannose glycans. Uromodulin is one such case. So it's this really cool protein that we make and secrete into our urine. And basically what uromodulin is it's a high mannose net that we throw over bacteria to trap them, and then we pee them out. And the way that works is it forms these very long fibers. You can see the fibers of the uromodulin protein here. And it's a heavily glycosylated protein. Most of the glycans are complex, but if you look here, you see this one glycan that always comes out as oligomannose, man 5 through man 9. Always. And the reason that happens is because that glycan at position 275 is buried in this little cleft in the protein. right? So rather than neighboring glycans providing steric restriction, it's the three-dimensional structure of the protein that provides steric restriction and prevents processing by minosidase 1. And so this particular glycan always comes out high mannose, every time. So we wanted to ask the question, can we take these all-atom design methods out of the Baker lab, use them to genetically encode high mannose glycans, and make better vaccines, right? And so Meg is the graduate student in the lab that took this project on, and Joel Allen came over and visited us at the IPD for about 6 months. Joel is in Max Crispin's lab at the University of Southampton, and they're one of the premier glycobiology labs in the world. So Joel came and taught us glycobiology, and we taught him protein design. And so the hypothesis here was, if we encode an exposed N-linked glycan, it will be processed by midosidase, it'll come out complex, it won't bind MBL, the immune system won't detect it. If, in contrast, we bury the glycan in this little protein cleft, minosidase processing will be prevented, MBL detection goes up, the immune system will detect this. And this is only possible, again, because of these all-atom methods that have been coming out recently. So 3 years ago, we couldn't model glycans, so there's no way you could do this. Now we can model glycans. This isn't a glycan, this is NAD, but you can model glycans and other post-translational modifications using these all-atom methods. So where Meg started was she took these super stereotype designed helical repeat proteins. You can see this thing is just pairs of helices added in layers. There's no twist, so you can just like add or subtract layers of helices at will. She installed an N-linked glycan in the middle of that V, and then we filtered these things down. We designed sequences and filtered them down, again, using structure prediction. And here, what we found was that designed steric restriction does indeed genetically encode oligomannose. So if we remove kind of the ridges on the side here and expose the glycan, we get a series of complex glycans that come out. You can see that in the mass spec. It's very heterogeneous. If you add back even a little bit of steric restriction, you already get to a mixture of oligomannose glycans. And if you add even more steric restriction, you not only go all the way up to MAN9, but we actually still have one of the 3 glucose residues attached. We've introduced so much steric restriction that the enzymes can't even get the glucoses off. So we've got MAN9 plus glucose. So that works. The hypothesis works. But using those Lego-like standardized proteins is not very generalizable. It worked in that one case, but that's just a DHR. We wanted a method where you could do this anytime, over and over again, in any protein structure. So Meg teamed up with Rohit in the Baker lab to develop diffusion so that we could design these things and generalize them. And so what you do here is you put a glycan into diffusion and just diffuse a protein around it, right? Use ligand MPNN so that it's aware of the glycan to design your sequence, and then filter by structure prediction. At first, when we did this, we just did unconditional diffusion, and we got almost entirely complex glycans. And this makes sense because you can see that the glycans are very exposed. So again, pink is complex, green is oligomannose, and it was almost entirely complex. And we did this a lot. This is one plate. We did like 10 or 11 plates. So Meg and Joel designed hundreds of de novo glycoproteins. We think these are the first ever de novo glycoproteins, but they're almost all complex, right? Like this one guy is oligomannose, but it's actually mostly unoccupied. The glycan isn't even in there in the majority of molecules. We tried a bunch of different motifs to feed into diffusion. We tried a bunch of different sequences. Almost everything came out complex. So then you go back to the code and you say, okay, that didn't work. What we need to do is we need to actually make sure during design that we're burying the glycan. So we used RASA conditioning, relative accessible surface area, which is a per atom score that you can use. It's a potential that you can use to tell diffusion, hey, I want the red atoms to be buried. I don't want them to be accessible to solvent. Blue atoms, fine, but not the red ones. And that turned all of these exposed glycans into buried glycans. And now we can design at will de novo glycoproteins with oligomannose glycans, so we can encode not only the position, but the composition of the glycan. So that's super fun, but do they do anything, right? So we wanted to use urinary or uropathogenic E. coli and urinary tract infections as a test here. So the way E. coli climbs up the urethra and gets to the bladder is by binding to genetically encoded high mannose glycans on a protein called uroplakin on the epithelium. It does this using this fimbrial tip adhesion protein called FimH. You can see it binds mannose right there. So we wanted to see if we could block receptor binding here with our de novo high mannose glycoproteins, which could presumably prevent UTIs. So this is a receptor blocking assay where we're getting inhibition on the y-axis. And you can see we tested a series of de novo glycoproteins where MAN5 provided really potent inhibition, whereas the higher mannoses, MAN8, MAN9, didn't do quite so well, and a control complex glycoprotein didn't inhibit really at all. This matches the known binding preferences of the FimH fimbrial tip adhesin, which prefers to bind to MAN5. So that makes a ton of sense, and we can actually block receptor binding from UPEC. We also wanted to make sure that these glycans could still be sensed by the immune system, and so we did an ELISA using MBL, mannose binding lectin. And here we saw the opposite result. Mannose binding lectin actually is known to prefer the higher mannose glycans like MAN8 and MAN9, and that's exactly what we saw here. And when we took our negative control complex glycoprotein called AP5 and we produced it with chifenensine to get exclusively MAN9, we got great binding by MBL. So it all makes sense. And so where we're going with this is we're designing what Meg and Joel are calling de novo nanoparticles that can be designed to optimally engage the immune system and complement and presumably make better vaccines. In order, I would say we've done proof of concept here. In order to turn this into an actual technology, we need to miniaturize the motif. You don't want to have to design a 300 residue protein just to get a mannose, right? So let's go to the smallest motif possible so that it's portable and we can put it in whatever protein we want. That's one of the things we're doing right now. And then there's the whole rest of glycobiology that is now accessible to these all-atom methods. And we had a rotation student this quarter in 5 weeks crack the next glycobiology problem, totally different than high mannose. So I'm super excited about this space. So I want to thank the people who did the work. I tried to highlight them as we went along, but here are the key players again, and our wonderful collaborators. Almost every project we have in my lab involves collaborators. We love working with other people. You learn from them. You can go further than you can alone. So love to collaborate. Happy, of course, for all the funding. And one thing, the movie doesn't work. Sorry, there's supposed to be a fun movie there. But one thing to say is we're growing. So there's a ton to do. These methods are allowing us to do things we couldn't do before. We're making molecules that, as Chris said, are going into clinical trials. If any of this sounds interesting to you, please reach out. So thanks for your attention. Happy to take any questions.
So for questions, I'll ask everybody to please line up at the microphone over there so we can get the questions recorded because we'll post the talk online. And maybe while folks are lining up at the microphone, I'll take the opportunity to be the host and ask the very first question. So super exciting to see the ability to control the types of glycans just with burial. Just wondering, it seems like it just worked, but in my mind is immediately thinking about the trade-off between burial and detection.
Yeah.
Did you have to actually model at all to see that you were keeping just enough exposed for the mannose binding proteins to be able to come in?
It's a great question. So we did not keep that in mind. We didn't have computational metrics to filter for that. We did when we were looking at designs kind of say, okay, does this look like it's still going to be accessible? But we didn't filter for it. And the key was just making enough of these things that we could have multiple points on that line. But now that we know, right, now that we've done that, we can go back and more intentionally design it.
Yeah, awesome.
Cool. I have two questions. One about, I'll ask the first one about the nanoparticles. Have you tried just putting the uromodulin high mannose glycan epitope like in one of these nanoparticles, and you can kind of just make that a known high mannose glycan and mix it with like the antigen so you have a hybrid nanoparticle, I guess?
Yeah, so that was actually the first thing that we tried when we started, is we took the motif from uromodulin and tried to diffuse around that. And eventually we got to the point where we were like, well, we don't want to be restricted to that. Like, we'd like to be able to do any motif. And so we broadened out and ended up getting hits from a bunch of different motifs. And I think this is a key point about the field of protein design generally, which is if you limit yourself to what exists today, then you limit yourself to what exists today, right? The whole point is, can we build methods that allow us to build things that don't exist today?
But did you try making those nanoparticles and like, were they high nanos, I guess?
So I actually don't know whether we got any hits from the Euromodulin motif. We got hits from several different motifs, but I don't recall if any of them were from Euromodulin specifically.
Yep. And then the second question, this is the de novo designed nanoparticle, I guess, configurations. It's like, you showed that you can design nanoparticles to show any antigen that you want, but I think that's maybe slightly different from the schematic that you showed, where like you want to position the antigens in a perfect configuration so that like an antibody can bind at specific parts.
Yeah.
So have you kind of gone in that direction?
Yes.
As well?
Yes. I just didn't have slides on it today.
Okay.
Yes, but that works.
It seems like a harder problem.
Yeah. Yeah, it's a fun one.
Thanks.
Yep.
Great talk. So I have a question. Maybe I missed this, but I was interested to know like why these nanoparticle forms of antigens could be outperform the soluble form of.
Yeah. So I think that goes all the way back to like slide 3 with the antigen receptor clustering and the improved trafficking in vivo. Those are really the two main mechanisms that we know of that give you better immune responses against particular immunogens.
Gotcha. Oh, I have a second question about the when you design these de novo sequences for these de novo nanoparticles. I guess like do you think about the immunogenicity of these amino acids and how this could bring into. Is it gonna be good for like multiple doses or?
Right. Yes, great question. So we always get antibody responses against the nanoparticle scaffold. We always measure those, we publish those, we're very upfront about it. Next question is, does it matter? And the experiments that we and others have done suggest that it does not matter. And specifically, what we've done is we immunize animals with bare particles that don't have any antigen on them. And so you jack up super high titers of antibodies against the scaffold. And then you come in the face of that pre-existing immunity with the same particle with an antigen on it. And it turns out if you measure the antigen-specific immune response, you get the exact same response whether you have the pre-immunity against the scaffold or not. So there is no deleterious effect on the antigen-specific response. And we've done that experiment, Icosavax did that experiment, the VRC did that experiment, and everyone got the exact same result. There are actually immunological reasons to think that it might improve things, because you get immune complex formation against the underlying nanoparticle, and the FC regions will engage complement and traffic the immunogen to the lymph node. But we haven't seen an improvement either. It's just been neutral.
Gotcha. Thank you.
Yep.
Hi, my question is on the involvement of T cells in the response. So, for example, one difference that you might get from membrane-bound versus nanoparticle presentation is the processing and T cell help that helps to isn't required in fact for the antibody generation. So, is that an area that you investigated at all? Is it?
Yes.
I mean, one extension of that is it important to incorporate like known T cell epitopes within your designs?
Yeah. So I didn't show it. We analyzed the T cell responses from the mRNA launched nanoparticles and compared to membrane anchored and everything. And we did not see big differences in either CD4s or CD8s. So you get basically the same T cell responses that you would get from membrane anchored, which I think makes sense. And then I didn't show it today, but yes, we have developed methods now where basically what we're doing is building the particles out of T cell epitopes. Because if you're going to build a nanoparticle to repetitively scaffold the antigen for the B cells, why not just build it out of T cell epitopes, right? And it's like a ligand efficiency sort of question. And that works too. So Sam Tips is the graduate student in the lab, and he's writing up that paper right now. But it's super cool. So you can, and it doesn't have to be a T cell epitope. You can incorporate any sequence you want into your nanoparticle scaffold. Yep.
My question got taken, but another kind of softball question here from industry folks. You know, this is a bunch of really cool new technologies that you've actually been able to push into the clinic. So when do you kind of know when here's a cool new technology, here's a paper, there's probably a ton of further optimization I could be doing on this? When is good enough to go to push this forward into a clinic?
I'm going to give you a very academic answer. As soon as you can convince somebody to do it. Right? Because like you always hear about like the academic valley of death, right? Where you're like, oh, I have this cool new molecule, and then people are like, yeah, not interested. So it can be very difficult to convince people to actually put in the resources to optimize, develop, run the clinical trial. I mean, it's a huge amount of work, right? Discovery is just the first part of this whole pipeline. And so it's really, you just have to have compelling enough data to convince people that it's actually worth putting the resources in. And that, the threshold is different for different indications and for different groups that might want to fund it, right? So like we do a bunch of work with the Gates Foundation, for example, and the Gates Foundation is doing global health targets that nobody else will fund. And so they need good candidates to go attack these problems that are market failures, right? And so you need good enough data to convince a funder like the Gates Foundation that it's actually worth, the X million dollars and everybody's time that it's going to take to go evaluate this thing clinically. So we work really hard to put together those data packages, right? Ask all the questions that funder or that company is going to ask so that we can de-risk the technology sufficiently to convince them to build a bridge over the valley of death.
I have a cage design question. Yeah. So first off, I think it's an incredible achievement that you can now build the de novo components to build the cages around.
It's a long cry from 2010, right?
The question is, how far away are we from getting away from the symmetrical docking and being able to fully diffuse cages? Are you working on this?
Yeah, and in fact, so that's already done. So what Sam has done, building particles out of T-cell epitopes, doesn't involve docking at all. So it's particle diffusion. It's still not easy. And I think the original diffusion paper kind of tricked everyone. It certainly tricked us, because there was one particle in there, HEO232, right? And it was like, oh, wow, you can just diffuse particles. No, you can't, right? And like, we tried, I think many people tried, and it just doesn't work. With the newer methods, it does work. It's still not easy, but it's getting better all the time. Actually, a huge problem there is prediction. You can't predict nanoparticles, right? And that is a serious limitation. So I'm bugging Frank all the time. I'm like, Frank, when can we predict particles? So we'll get there soon.
Okay, we'll be watching.
Yep. Cool.
Fantastic. Well, no more questions. Thank Neil again.
Thanks, guys.
And thanks everyone for coming out today. I know this was a bit of a short notice, so really super grateful to have the crowd. And please feel free to grab some more food and drinks on your way out. Snap a selfie with the Citgo sign that's being disassembled, and we'll